Hello everyone, welcome to another session of Luminavus online conference on light pollution. Today is the second day and we will launch it with Dr. Svenja Tidau. First, I would like to thank Dr. Tidau on behalf of SEO, the organizers of the event, and all of Luminavus partners for her participation. Dr. Tidau's research is focused on the sensory ecology of marine organisms, and specifically how the anthropogenic pollutants like noise or light impact their behavior and development. During her PhD, conducted at the University of Plymouth and at the Galway Mayo Institute of Technology, she assessed the behavioral impacts of anthropogenic noise on the European hermit crab, showing that noise affects these organisms across sensory modalities and impacts their behavior at both individual and group level signal and natural cues transmissions. Currently, she is working as a postdoctoral researcher for Project Aves. Aves translates into artificial light impacts on coastal ecosystems. It's a project funded by NERC and developed by Professor Thomas Davis at the University of Plymouth. Here, the team is conducting laboratory experiments to determine the impacts of artificial light on different aspects of coastal organisms and coastal ecosystems. Dr. Tidau is also a member of the Association for the Study of Animal Behavior, the British Ecological Society, and the Marine Biological Association. She will now present the talk titled Artificial Light Impacts on Coastal Ecosystems. Thank you very much, Dr. Tidau.
Hello everyone, my name is Svenja Tidau. Thanks a lot for your time to listening to my talk. My talk is titled Artificial Light at Night, an Underestimated Driver of Global Change in Coastal Marine Ecosystems. And I'm going to present to you the current evidence that we have of ALAN impacts in marine and coastal ecosystems, and I will identify 10 pressing questions that still need to be addressed and the potential to do so. I will also, towards the end, introduce my team and give you a few examples what we are currently working on. Before going into the details of light impacts on marine ecosystems, I want to give you a quick introduction on the biological adaptations to light and darkness in marine ecosystems, for you to better understand the background and why ALAN could have potentially really profound impacts. As for land, the main light and dark cycles govern marine ecosystems. Those are diurnal light cycles, lunar light cycles, both of which vary throughout the year and between years. But in marine ecosystems, we have two other components that are really important to consider. For coastal regions, that is, for instance, tidal cycles and tidal attenuations, because it means that organisms are up to twice a day completely exposed to direct light, whereas twice a day they can be covered. Another aspect is really important to keep in mind is that the properties of seawater, because of seawater has a lot of different metals and organisms in it. Light will be attenuated in terms of its intensity and spectra throughout the water column. So light levels at the surface of water are very different to what an organism might perceive throughout the water column. There are some really fundamental adaptations of organisms to light in marine ecosystems. And to introduce all of them would be too much, but I would like to give you two really important examples. One of which is probably very widely known. It's a coral mass spawning that is timed around the lunar cycle. And basically, corals release eggs and sperm in a synchronized time period in order to maximize their reproductive output and their reproductive success. And while this is very known for corals, it's actually a process that occurs across many marine organisms, many marine invertebrates. And that is for the same idea, so that they can maximize their reproductive success. So for instance, in temperate regions like the UK, like shore crabs, for instance, would also synchronize their reproductive events. And these are not the only processes which are really important, but there's also something like style vertical migration that occurs in zooplankton. And you can see that example here on the slide. So what zooplankton does is that during the day, they are deeper down in the water column to avoid their visual predators. And in the night, they tend to migrate towards the surface to forage themselves. But what you can see here on the graphic, which is based on a study by Ludwigsen et al., is that even moonlight is suppressing the migration of zooplankton. And what that means is that these organisms can detect really dim light conditions quite deep in the water column. So first, we should have a look at what we know about ALAN and how it is distributed, and if it actually is a global phenomenon. And here you can see the FARCI map, which shows you the sources of land-based light pollution throughout the globe. And on the right-hand side, you see a cutout of this map for the target region of this conference. Basically, the more yellow or red an area is, the brighter these areas are lit up at night by artificial light. And indeed, a study from 2014 kind of narrowed this down for what it means for the global coastline. And we can see that 22% of the coastline is exposed to artificial light at night. And there's probably a lot more by now. There are different sources of ALAN that marine ecosystems are exposed to. So here I found some examples from the Canary Islands, but this is something we can find across the world, that often shores are heavily lit because of promenades of housing, because of ports and harbors. And all these lights, as you can see on these images, are actually spreading out into the sea. Other purposes for lighting, like this recent example from Plymouth, is it might be for artistic purposes. And these are land-based coastal sources of light pollution, but there are also offshore sources of light pollution. So here you can see a few examples. For instance, there are gas and oil platforms in the sea. There's a lot of ship traffic going on. And then actually humans are exploiting the fact that animals are attracted to certain types of light at night. At the bottom of these images where you see Southeast Asia and fishing for squid, where fishermen use green light to attract fish and to enhance their catch. So while there are coastal sources and land-based sources of direct lighting, there are also sources of artificial light out in the sea. But there's an indirect form of light pollution that is potentially a lot more important for marine organisms, and that is what is called artificial sky glow. If you have been on a nightly walk and you look towards a city or even a town, what you will often have noticed is a dome-like shape above a city or town. And that is direct light that is emitted into the atmosphere, scattered there, and then reflected back. And while this might seem very dim for us, and indeed it is really dim, so if we compare the intensity, the brightness from a human perspective, direct lighting has between 10 and 100 lux, and sky glow is 100 times dimmer than that. Why it is potentially so important is because the natural sources of light that animals have adapted to, like the full moon or even stars, which they use as a cue, are well masked by artificial sky glow. And in addition to the fact that sky glow can mask those natural cues, it also expands hundreds of kilometers away from its source, and there was potentially also out to the sea. And that brings me to the first knowledge gap, which is basically that we have a very poor understanding of how ALAN is distributed underwater and how much there actually is penetrating through the water column. In order to better understand that, I first want to give you a quick example of natural light and how it penetrates down the water column. So what you can see here on the top panel is sunlight, and you can see that it is broad in wavelengths, so it covers all colors of light. But if you then look at the panel below, you will see how daylight penetrates down the water column if we are offshore, which is usually water that is relatively clear. And what we can see there is that particularly blue blue, a bit greenish light penetrates best down the water column. The bottom panel shows you an example for coastal waters, which, because of its properties, will have more suspended matter, for instance, and so the spectra that travels best in the sea is a bit shifted towards the green-yellowish, and that also is what we often perceive as humans when we look out in coastal waters. That can be really green, yellow, sometimes even really murky brown. And these characteristics also have an impact on how Alan is traveling through the water column. So here's an example. Indeed, it's the only study I'm aware of where Alan has been measured underwater. And that is a study that has been conducted by Tamir et al. In 2017 for the Red Sea. And basically what I want you to look at mainly is the yellow and the blue dots. So here they measured the intensity of light and how it changes depending on the spectra and with depth. And what we can see is that yellow light, even though more intense at the surface, gets weaker really quickly with depth, but the blue light doesn't lose too much of its intensity and actually is the most bright light down in the water column. And why this potentially really matters for marine ecosystems is that with the rollout of LEDs, because of our intention to save energy, we introduce more blue light into the environment. So below this lamp, you can see the spectral range of a typical LED, and you can see that it peaks in the blue spectrum, which is the wavelength that travels best in most marine ecosystems and best through the water column. And that means that potentially ever more organisms are exposed to Alan at greater depth. What we then did in a recent study led by Thomas Davies is that we wanted to better understand how Alan is perceived by organisms and how much Alan, which is biologically relevant, is reaching the seafloor. When I talk about biologically important or relevant, I mean irradiances that can elicit a response in an organism, because whether or not light can be perceived depends a lot on the sensitivity of an organism. And we used as an example copepods because we know their spectral sensitivity across the blue, the green, and the red spectra of light, because it has been assessed before. And these organisms are really widespread. I'm now going to show you two maps, and they can be a bit intimidating at the beginning, but I will talk you through them starting from the bottom. So what we've done, we wanted to know how much of Alan is actually reaching the seafloor. And the bottom panel shows you the Alan intensity right on the surface. And basically, the lighter it is, the more light you find at that part of the water column. So not surprisingly, at the surface of the water, it's very bright, and the light is very bright in all the three spectra: blue, green, and red. Then the second from the bottom shows you the light intensity right below the surface, and you can already see that the light is getting darker. The third from the bottom then is the light conditions as you will find them during low tide. So basically, this is Plymouth Sound, and we have two tidal cycles. And what you can see is, particularly for the blue, that only the bay there towards the left-hand side is still lit with Alan, so that's the Alan that will reach the seafloor at that location. But what is important here to point out is once we reach high tide, so if we go to the top panel, we will see this is way darker. So basically less Alan is reaching the seafloor. But where it becomes really interesting is that this is only for the blue. If we look now on the panel with the green wavelength, we see, not surprisingly, that because in coastal waters, green light penetrates best to the water column, that substantial intensity of Alan is still reaching the seafloor even at high tide, because it's really important whether we measure blue light, green light, red light, or just all the spectrum in once. So what you see basically is that during high tide, in Plymouth Sound, a coastal area, green light penetrates still down to the seafloor. This is the example for clear water, for clear sky, but because Plymouth is in a temperate region where we often face clouds, it's really important to consider clouds as well. And basically, rather than talking through the whole panel, again, I want you to mainly focus on the green wavelength and on the top two panels, which is for high water and for low water. And what you can see here, if you just compare the green wavelength for high water and low water, is that clouds really amplify the intensity of ALAN that's reaching the seabed. So areas that were previously dark, once it's cloudy, and it's quite often cloudy in Plymouth, as I can assure you, a lot more ALAN will reach the seafloor and is detectable by copepods. So I've shown you the two studies that have assessed how ALAN is distributed in marine ecosystems. What I now want to focus on for the remaining talk is mainly on the impacts that ALAN has on marine ecosystems. And what I've done here, I've been looking at all the studies and were published in peer-reviewed journals, and I've been looking at the spatial scale of the response that they covered, and I've been looking at the biological rank. So looking whether a response on the cellular level has been assessed, organism level, and up to ecosystem level. And I've grouped those studies based on their phylogeny, forming really broad taxonomic groups. What I want you first to focus on here is the level of organisms. And as you can see, that's where we have the best understanding. So that's where what most studies have assessed. That's the response that organisms show to ALAN. If you look on the population scale, you will see that we're shifting in scale, so we get a better large-scale understanding. But what also becomes really apparent is that few colors really dominate. And one is the kind of turquoise green color, which are studies on seabirds. And then there are these brown dots, which you will also find a lot on the organism level. And those are studies that have been looking at responses of sea turtles to ALAN. You will find this one dot up on the right-hand corner, which is the one study that has assessed ecosystem impacts of ALAN. And if we then go further down to the microscale of cell physiology and development, you will see also way fewer studies than we have on the organism population level. And I will use this chart and talk you through all the different biological ranks and the state of knowledge that we have on each of these ranks now. So the first, or the second, I should say, research gap that can be derived in this talk, the first from this graph, is that we have a really strong taxonomic bias in our understanding of ALAN impacts. And that's largely driven by research that have been conducted on seabirds and turtles, because both groups of species, they breed on land, or they spend the remaining time of their life to a large extent at the sea. Then we have a fair amount of understanding on the impact that ALAN might have on fish, accounting for 19%. But if you then see that another quarter of studies covers this whole broad range of marine mammals, invertebrates. So what do we know on the level of organism response? We know quite well how it can change activity patterns, and that's not surprising because stay active animals will be more active, whereas nocturnal animals will reduce their activity. We also understand and have shown in many studies that ALAN can lead to disorientation. And again, this is where really studies from sea turtles and seabirds have also really been paving the way for this field to really show that ALAN is a pollutant that we should focus on more. But there are many behavioral contexts where we don't really know what the impact of ALAN might be. And here I've pointed out some which are also related to population and reproduction, which are major in courtship and parental care. But of course, there are many more behavioral contexts. On the level of populations, again, leading studies on seabirds and turtles, We know that ALAN can have impacts on reproduction and most of all on offspring survival, because it can lead to mortality if an offspring gets misguided and doesn't find its habitat, or for instance, if seabirds are grounding. But there are also studies on other organisms, like here the clownfish, that have shown that if eggs of the clownfish are laid under ALAN, then 0% of will hatch. So basically, zero reproductive success. And there are many organisms, as I already outlined in the beginning, that are timing their reproductive events around moonlight. So there's a lot more potential for other organisms to be impacted in a reproduction by ALAN. And while we do know a bit about reproductive timing and offspring survival, what we really don't know how many of these offspring are actually recruited into the population. So how many will be able to reproduce themselves the next year and therefore contribute to the gene pool. The whole question of recruitment and population dynamics is one that needs also more work. We want to know what the ALAN impacts are on the community level in coastal and marine ecosystems. We know a fair bit about the effect of ALAN to alter the composition and abundance of species, that it can alter the use of niche and trophic interactions. Again, not surprisingly, fish and diurnal foragers, they seem to benefit, whereas their prey obviously is in the disadvantage because it's more heavily preyed upon. Nocturnal foragers can be suppressed in their activities, and here is a study from Rodríguez where I've already shown you part of the panel. And what you can see basically is that in the presence of ship light, that zooplankton will suppress its divertical migration, and that this response holds true down to 100 meters of depth. Another example is that when looking at trophic cascades during day and night, and then observing trophic cascades during artificial light at night, you will see that the interactions and that the behavior of fish and preying intensity resembles more the one you would see during the day. And that this is actually also altering the substrate assemblages on these ecosystems. But there are also more questions to ask on the level of communities. So we know that in some ecosystems, there are certain species which are particularly important because they can be ecosystem engineers. And there are other questions to be asked, such as how is symbiosis or competition altered by ALAN, and really understanding better the mechanisms behind the patterns that we see. So that kind of brings me to a little summary so far. And what we can say is that overall we have a bit of an understanding of what's going on in terms of organism, population, and community responses to ALAN. But if we look at the all of these spatial temporal scales, we're really lacking insight into ecosystems and into the sub-organismal level. And what we're lacking in ALAN research on marine ecosystem completely at the moment is what are the long-term impacts. So is what we observe mainly in acute shock response of organisms, that they're repelled by light or attracted to light, or can this actually translate into long-term changes? So what do we know about ecosystems? There's one study by Albrecht et al. From 2008, and they've been using remote sensing and spatial analysis tools to show which coral reefs are particularly exposed to ALAN. And while this is very important to assess potential threat, it does not tell us anything about how corals might respond to ALAN. Two relatively recent lab studies, and actually a few more since then, have shown that corals undergo oxidative stress when they're exposed to ALAN. Their photosynthesis is impaired, and when looking at gene expression, we can see that genes are differentially expressed, and those are related to the health of and the growth of corals. But we don't know yet how these two link together in terms of what does it mean for the ecosystem functions and services of corals. And of course, corals are one example, but there are a lot more habitats that deserve to be looked into. So for example, the dive vertical migration that I mentioned before of zooplankton is really important because they form part of what's called the biological pump of the oceans. So while zooplankton migrate up and down the water column, they also help to transport nutrients from the surface and down to deeper layers. What is also important that right now we have some key habitats and key species that we've been focusing on, and that makes sense because they can present themselves as really good model organisms and systems to start understanding island impacts, but there are a lot more habitats that need to be investigated. And for instance, what's really also challenging in terms of technology is understand better what is going on throughout the water column offshore. And this is also tricky because research generally is really hard to do offshore and financially really, really difficult. Another level that I think should be particularly looked into is looking into how organisms processes are affected on a cellular and physiological level. I mentioned these few studies on coral responses, but what that also shows is that so far only really few biomarkers have been looked at, and we have few studies and few taxa where we have just a bit of an idea of what ALAN might mean on that level of response. But there's a whole suite of biochemical indicators that have been used to look at other stressors on marine ecosystems. So there's huge potential to apply those into an ALAN context. One of those tools or approaches and perspectives is to look at the chronobiology, because chronobiology is a well-established field, and we know from many organisms that they have circadian rhythms or that they show monthly rhythms. So a really important next step for ALAN research would be to look more how these processes are affected in marine organisms. There have been a few studies on terrestrial organisms, but not for marine organisms. And then on the level of development of organisms, there are a few studies that you maybe remember from the plot that I've showed you, but most of those studies were based on aquaculture. And what the problem there is for us as biologists, if we want to translate our findings to what ALAN impacts means in the real world, is that those light levels are often way above what an organism would naturally experience. So it's really hard to translate the responses that we see on the level of development of organisms into the wild. I want to point out what I hear in this presentation make my last really important research gap, which is the impact of artificial sky glow. As I said in the beginning of this talk, artificial sky glow is really important to look into because it can extend light away from its source hundreds of kilometers out to the sea. And it is important because marine organisms are highly light sensitive. So this could have potentially profound effects that we're currently not really looking into it enough. We published a study this year in Current Biology, which was driven by Daniela Torres. She was a master's student at Svenja University at the time. And I want to give you a bit more detailed insight into her study. So for the experiment, we used the sandhopper Talitris saltado as a model organism. And what Talitris does, it's buried in the sand during the day, and it comes up to the surface in the night to forage on stranded algae. And that is a really important process because while it's foraging on the stranded algae, it also recycles nutrients. It breaks up the algae and makes these nutrients available for other organisms. So here you see a basic drawing of Daniela's setup. And what we were looking at in the study was looking at the probability of sandhoppers migrating from where they emerge in the sand to the food source. So the probability of doing so and the direction of their migration if they would migrate. And Daniela has been in the field on many nights over 4 months and has been altogether looking at 960 sandhoppers. So the first finding of our study is that sky glow reduces the migration probability. So basically, sandhoppers will practically not migrate anymore. And what I should have mentioned in our study is that we also took into consideration 2 more factors besides sky glow. We were accounting for full moon and new moon, and we were accounting for cloudy and non-cloudy conditions. So the first 2 panels show clear conditions, and the two last ones show cloudy conditions. And what we found is that in all of these treatment combinations, there's a reduction of the probability to migrate in sandhoppers in the presence of light, which is the lighter of these 2 bars here, of the 2 gray bars. The only condition in which we did not find an effect of sky glow was during full moon and cloudy nights. The second question we had was sky glow alter migration pattern. So again, we had the same treatment combinations. Here you see the results that we observed without sky glow being present. And what you see here are the red lines that point out the expected direction that Tilitris should migrate. And what we can see here at the bottom panel is the observed direction. And what you can see on the second plot from the left, the one under new moon, is that the migration pattern of Tilitris was opposite of what it was expected. So that suggests that Tilitris might have perceived sky glow, the artificial sky glow that we introduced as a full moon. So now, after presenting you the evidence that we currently have and the knowledge gaps that we have, I would like to introduce our team to you and give you a bit more outlook of the current project that we are working on. The project I'm working on is called Alice, which is Artificial Light Impacts on Coastal Ecosystems. And we are a team of biologists, ecologists, and optical oceanographers wanting to combine the biology and the physics of light pollution in our research. So we take different approaches, and we conduct experiments in the lab where we assessing different responses of coastal organisms, very common in temperate regions. And then we want to feed this into models and mapping together with understanding better how Alan is distributed underwater. So on the side of the experiments, and that's where I'm mainly working on, our aim is to assess Alan impacts on really critical life stages, such as larval development, larval mortality, and recruitment into the population. Another focus of ours is to assess species interactions and looking at predator-prey interactions, but also in competition of species. And then here on the right-hand side, you see another example that we're looking into, which is the potential for mitigating Alan impacts. So what you see here is our lighting setup, and you see an LED, and hopefully it looks a bit more yellow to you than the white LED you're used to. And that is because we've applied a filter and we want to look into the potential of filtering out the blue wavelength of LEDs. What is also important to look into here, to pay attention to, is this little round bowl in the center. That is our moonlighting system. And that's something that we believe is currently lacking in most lab experiments, that we really need to simulate properly the natural nighttime light conditions in order to contrast them with Alan. So we've developed a moonlighting system in order to be able to do so. Second set of approaches combines modeling and mapping, and this is really where the biology also meets the physics. So I've already shown you one study where we looked in the Alan distribution and how it is perceived by organisms, and that was on a regional scale. But ultimately, we really want to scale this up on a global level to better understand where are the hotspots of ALAN exposure throughout the water column at which depth. That could then also help, for instance, to inform biologists and ecologists to target certain regions and to look how those regions are already affected by ALAN. What we also do is, here you see a life cycle of an oyster. By looking at the lab on these different stages of a life cycle of an organisms, we want to bring them together in doing population dynamic models and better understand what the observed responses we see in the field actually mean for populations, whether they are stable, decreasing, or increasing. And ultimately, if we can bring together the biology and the physics, it might also allow us to look into the biogeography, meaning that we can look into regions that are highly impacted for certain species, and whether or not species might maybe able to shift their habitats in response to ALAN in order to avoid it should it be detrimental. So now I would like to sum up the presentation and really stress that in my eyes, ALAN is a potentially underestimated driver of global change in coastal and marine ecosystems. And this is so despite the fact that we currently have quite a few knowledge gaps, but what we know how natural light is used, there's huge potential, and we need to better assess what the ALAN responses to of marine and coastal organisms are. So I really would like to stress here again that sky glow is a form of light pollution that extends far away from its source and has the potential to mask natural light from the lunar cycle. And we've shown that sky glow has an effect, for instance, on Tilitros, and that is just a model organism. There are a lot more organisms where we know that they use the moon, so we need to look better into that. Another fact that makes ALAN a potential driver of global change is that we are rolling out LED use. And that has many advantages for us humans and for our energy consumption, but we also have to look into the potential side effects. And in the case of marine ecosystems, that means first will mostly penetrate deeper into the water column and reach more organisms. And we've partly shown this with the study of Plymouth Sound and how much light is reaching the seafloor, because Plymouth is a largely LED-lit city. But there's a lot more potential of responses in organisms and ecosystems we're not really aware of. And I just quickly want to summarize them with pointing out again the dive vertical migration in zooplankton, the spawning of corals, but also other marine invertebrates, that if it really leads to 0% hatching, like in the clownfish, that would be devastating for populations, and that we really have to look more into the genetic and cellular processes of how ALAN might alter them, because in the few examples where we do know something on this level within organisms, we do know that really crucial processes are altered. And that's what I wanted to end my presentation with. Thank you very much.
Well, thank you very much, Dr. Tidau. That was incredibly interesting. You covered a lot of aspects. It's very a global overview of ALAN impacts on ecosystems down to the organism level. I would like to know if it's possible to know the so how important is it to differentiate the impacts of direct lighting versus artificial sky glow, or if it's really a factor, because I would say that the solutions would be similar, but maybe the impacts of both light types would be slightly different. And how can we understand the different impacts, and if you can walk us through the solutions you propose as well for this kind of pollution. Thank you.
So I think first, you're right, they obviously linked artificial sky glow and direct lighting, because artificial sky glow is a consequence of a lot of direct lighting. So solutions to mitigations would be to reduce the lighting, to consider part-time lighting at night, to consider dimming. Something that's very important and could be done on the level of designing of street lights, for instance, is to improve shielding. So basically, rather than having light source that goes up into the air when we actually want to illuminate the streets, if you would apply shielding, you would also reduce potentially sky glow. So definitely the entry point to reduce light pollution is on the source of direct lighting and the lighting regime and the intensity of lighting, but also a really untapped potential is to look into using filters and see how organisms might respond to that. But obviously, so I talk from a marine biologist perspective. If we can filter out blue light, that would be probably having the strongest effect on marine organisms, but it might shift the spectra to light colors that are important for terrestrial organisms, but actually then have negative side effects there. So I think it's actually a really interesting question to the marine biologists and terrestrial ecologists should come together and see what one solution does for one habitat and how another one is impacted.
Thank you very much. I remind the audience that Dr. Tidau will now be available for 15 minutes to answer your questions and comments on her presentation. Thank you again on behalf of everyone. That was really, really interesting. Thank you.
Thank you.
Well, welcome all. I hope that you are enjoying the webinar about light pollution impact. And I'm very lucky to present, one of the biggest researchers in light pollution impact. That is Dr. Travis Longcore. Dr. Travis Longcore is a researcher for the University of California and Los Angeles Institute for the Institute of Environment and Sustainability. And he has been working in light pollution impact since the past 20 years. And like I said, I'm very excited with this presentation because he was the one that basically, and his article, Ecological Light Pollution and the Ecological Consequences of Artificial Night Lighting, that basically set the path to light pollution impact in ecology. And I hope that you enjoy the presentation, and I will pass to Travis. And thank you very much for being here. And let's hope that we learn much more about light pollution impact.
Well, thank you so much for that great introduction. I really appreciate it, Tanya. So I'll start here by sharing my screen and getting some slides here up on the screen. So what we wanted to talk about today was about the mitigation of light pollution through control of spectrum. And spectrum is actually a big topic for me right now. And it's not to draw any attention away from the brightness or the direction of lights in light pollution mitigation, but often there are public safety considerations that can't be overcome, and you have to deal with spectrum as your means of mitigation. We find this quite a lot in the United States. So thank you for having me here today, and I'm really looking forward to interacting with you after the talk. So, and really this does come about because of the increase of the use of LEDs for outdoor lighting, and then becoming a financially viable approach, because the color of the LEDs, especially early on in the technology, was very much dominated by blue light and full spectrum white light with a heavy emphasis in the blue. And that's because the LEDs themselves start out with a blue pump, the diode itself, and then have a phosphor that spreads the wavelengths into the longer wavelengths. And so there's a technological reason that we saw this huge shift when you go from some older technologies, especially high pressure sodium, into LEDs. And this has really become an issue of awareness in the industry as well. This is actually a spread from an industry magazine talking about how LEDs are affecting wildlife. And you can see it on the landscape. This is where I live, and not on this bridge, but the city of Los Angeles. And this bridge has been relit from high pressure sodium, the orange lights in the background, to about a very white, tending toward blue LEDs. And the whole city is being relit in this manner. And it's being taken as a point of pride by the city. And of course, this comes along with it perhaps some problems that we need to deal with. That is the Los Angeles River that's running underneath of that bridge, which is it's constrained in a channel there, but it is in fact a river. So the big topic that changes, the big thing that changes when you go from traditional high pressure sodium, low pressure sodium, and some other legacy sources of light is the color. And the way that the lighting industry has, and lighting designers and engineers describe color, is through correlated color temperature, which is goes from low numbers, 2000 there, that's 2000 Kelvin. It's on the Kelvin scale. It's as if you heated up a body, and as it gets hotter and hotter, it gets bluer and bluer. It's like the hot part of the fire is blue and the cool part of the fire is yellow. The same thing here. So a low correlated color temperature, or CCT, is maybe 2000 or even 1800 or lower, and then a very high one would be 6,000, 7,000. But even in fact, 5,000 is quite a high color temperature for outdoor lighting. You can see how blue that gets as you get farther over to the right there. So we talk about these things in terms of correlated color temperature. However, there are a lot of criticisms about correlated color temperature. We may end up in the future having some other metrics, and so I'll talk about that a little bit. The other ways that we might measure impacts other than correlated color temperature as standing in for how sort of yellow or blue a light is. And ironically, a low color temperature is called warm because it's yellow, when we think of yellow as a warm color, and a high correlated color temperature is cold, which, because it's blue and we associate blue with ice and being cold, but it's actually the correlated color temperature is higher. So I want to talk first a little bit about kind of what naturally happens as we head into the nighttime and just how different it is. And anyone who lives on this earth outdoors and sees it knows that light changes as the sun goes down during a sunset, and that there's a whole suite of things that go on at night that are just simply different. And so this is actually the correlated color temperature out the window that you're seeing in the background right here in an evening, looking at the temperature of basically the sky, very blue sky, so that's a high color temperature. But as you get into the evening, and we're going through sunset here, the correlated color temperature goes up and then it goes down again. So it goes up, it gets blue, and then it goes down and it gets more yellow. We get this golden look as the illumination goes lower and lower. So that's the sequence of a natural signal going into the nighttime. And in fact, some biological organisms like coral reefs and corals will actually respond to this change in the spectrum as opposed to the change in the illumination, because it's a very predictable change under natural circumstances. The bottom there is just the brightness as we go through that period. Everything, there's a whole life that sort of takes place after dark. Many species are adapted to doing things in the dark and the nighttime. This is a datura. It's a medicinal plant, a native plant here in California. Not my video, but it opens up only at the nighttime. It's pollinated by nighttime pollinators, and it sort of closes back down during the day. And so there are all these signals of it becoming nighttime that then trigger things in the biological world to occur like this. We look outside and what we see is that the world is getting darker and that the color drains out of it. And so on the left-hand side of this, we see that human color vision is at the daylight and into twilight. And so you're seeing that under natural light levels, and the rainbow here represents that humans are seeing color. And as it gets darker, we as humans, we see the world turn to gray as we get into just using our rods and our cones, maybe a little bit of color. And then we get down into when it's dark, we're only using our rods and we're only seeing shades of gray. And so the color drops out of the nighttime world, but it doesn't do that for every species. And nighttime lights can be bright enough to bring color vision back for organisms that are adapted to vision under those brighter conditions. But the thing is, the world doesn't go gray for everything. And here we have a number of insects, starting here with a honeybee and into a couple of moths over here on the right-hand side. And they have color vision that actually extends into far lower illumination than humans do. And some where actually they can see into dim starlight. They're perceiving color in the environment. So it is worthwhile thinking about color, even though from our human perspective, the world sort of has the color drain out of it. There are other species looking at that and seeing it, and I might also add, seeing other parts of the spectrum of electromagnetic radiation beyond that which humans see. So these differences end up being very important. Other things that happen is, birds go into a state of reduced metabolism. So that signal to go into the nighttime, and this is of course for diurnal species. Doves have about a 20% decrease in metabolism. Sparrows down 50%. The one on the lower left there is an oak titmouse. It's a small bird that actually lives in the oak tree outside my house here, and has about a 70% decrease in its metabolism, and then hummingbirds down over 80% into basically a torpor. And so there are a lot of these things that are going on every single night in response to these natural patterns of light and dark. There are physiological responses, there are behavioral responses, and of course what we're all here to discuss is that the world then changes when you illuminate it with electrical or other outdoor lighting at night. So this is a map from the wonderful lightpollutionmap.info that shows the most recent satellite data composite of upward escaping radiance across Europe and North Africa and into the Middle East. And the reds are the brightest, and you can see that this is a dramatically transformed landscape. And so the question here is, you know, the consequences of that, and then for me, especially today, how do we deal with that relative to color? We have this long history now to look at about when we put these artificial stimuli into the environment, how do species respond? This is an old painting of the Eddystone Light showing the attraction of birds to lighthouses, and we in fact know this for birds. There are accounts in newspapers back into the 1800s of attraction to light at night of birds, especially nocturnally migrating birds and seabirds. And we can then trace that research forward up into the current day. This is a one-night kill of migratory, nocturnally migrating songbirds in North America. It's a few years back at a communication tower where they're attracted to the lights of the communication tower, circle it, run into the wires, and then are found there on the ground in the morning, dead. And then, of course, seabirds are affected as well. It's perhaps even more so because of their particular foraging strategies and food cues out on the ocean. But we have as everyone here at this seminar probably already knows, the attraction of seabirds to lights, especially juveniles from their nests or fledglings heading out to the ocean for the first time, and efforts to repair that. And then they also act synergistically with glass. So this is I forgot, ovenbird that ran into a cabin window in the woods in Michigan. Which we were able to pick this one up and rescue it temporarily. It recovered, flew away quite happy, but the combination of indoor light and glass led to a lighting environment where this bird ended up having run into a window. And it's not just out in rural areas. We have bird glass collisions exacerbated by light pretty much everywhere where there are humans that build buildings with lots of glass in them. And traditionally, if you go back in the ornithological literature, you know, people as famous as Mayfield were writing, just don't really worry about all this stuff. Yeah, so birds are attracted to towers. They're killed in proportion to their abundance, and so, you know, this isn't really an issue. Now, this is quite an old position, and we've, I think, learned a lot more and adapted our understanding since then to realize that, in fact, some species are much more vulnerable to light attraction and mortality than others. And there are many species that we care about of extreme conservation concern that are affected by this that we should be worrying about and mitigating. And it is not something where we should shed few tears. We should, in fact, pay attention. This study just from a couple of years ago in Scientific Reports, looking at how light pollution is the greatest in the areas over which nocturnally migrating species have to go. And those nocturnally migrating species include many species of conservation concern. And this is just looking at the big migrations. This doesn't even start to talk about the seabird issues and how they are affected. So as I give talks about this to the public, and this is probably review for everyone here, it's like, well, what do you do? What do you do about this issue? And these are the sort of the 5 basic things that you can do for mitigation. Light only where you need to light. Turn the lights off when they're not in use. Sometimes that includes doing special events to turn lights off during high sensitivity times, lights out programs for migration, during fledging for seabirds. Use only as much light as you need, so the brightness matters. Direct the light only where it's needed. And then I summarize this as use the lowest possible correlated color temperature for the goal of your lighting. And here I'm using correlated color temperature because that's the standard metric that you can find on the box of a lamp and know when you buy it what that number is. But it's a shorthand, and we'll talk about what that shorthand means in a little bit. So the first four, I think, are pretty obvious, but not to be forgotten as we talk about the last one here in some more detail, which is the spectrum of the light and how we go about dealing with measuring that and manipulating that in order to reduce these impacts. So spectrum, I would argue, in some instances, not all, and there's active debate over the degree to which it is important, but is as or more important as intensity for some ecological effects. And actually, it's hard to disentangle spectrum and intensity because how bright an organism perceives a light to be is in fact correlated to the spectrum because we're used to maybe measuring light for how humans see it and measuring it in a unit like lux, which is weighted by the sensitivity of the human eye. But in fact, other organisms have different visual systems and see it differently. So 5 lux of a light to humans that is very red and 5 lux that is very blue, humans might see that the same, but to a mouse, which doesn't see into the red, might see the blue light as 4 times brighter than the red light. And that's an actual sort of intensity, not just they're seeing a different color, but the intensity that they perceive because of the photoreceptors in the mouse's eye is actually much brighter for the blue light than for the red light. And so that's why spectrum sort of goes hand in hand with intensity, and we can use it and perhaps in some instances to greater and lesser effect manipulate it in order to reduce impacts. What we're looking at here is actually an experimental setup in Germany, Gerhard Eisenbeis, looking at insect attraction. In the foreground, which you can't see at the bottom of that grassy slope, is a river. And he was measuring insects attracted to different lighting technologies. And out there you see high pressure sodium, which is the orange. There's a couple of LEDs. There's a metal halide. There's a number of different technologies here. And so he compared these in terms of insect attraction and found that the most attractive light to the insects was a mercury vapor. We don't use them very much anymore, but it was very attractive because mercury vapor emits light in the ultraviolet, and that's particularly attractive to most insects. Metal halide, also a lot of short blue and wavelengths, was the second most attractive. High pressure sodium, the orange that we've had around for decades, is below that, and then cold white and warm white LEDs, which are different from each other, less than the other sources, but there's that difference between the cold and the white is pretty significant. And this is something that we can take advantage of as we design systems for the future. And in fact, more research on insects sort of starts to open up the question of whether the high pressure sodium is better or worse in this regard. So the whole idea behind this is, as I mentioned, is that the visual systems of different species are different, and some see into some areas of the spectrum better or not at all than humans do. Because humans are always the focus of outdoor lighting at night, and we see across a certain spectrum that we're very familiar with, but other organisms, in this instance deer, like mice, don't see very well or at all into the orange and red. So one can imagine using spectrum then, if you wanted to reduce impacts on deer, using something that has those longer wavelengths of the orange and red and yellow and less of the shorter wavelengths. So basically using the area of the spectrum that humans take advantage of while avoiding those that your species of interest is seeing the best in. And that's the sort of the principle behind looking at that. But to do that, you need to know what those responses are. And that's where we've done a little and need to be doing more. And I know of a couple of groups around the world right now that are actively working on this. We have a curve here. It's a generalized curve for like all insects. Now, admittedly, not every insect species is going to follow this curve, but it's been tested on data from studies on, I think, 2 continents at least. Maybe we had 3 in there. published a couple of years ago. And what it shows is that the photoreceptors that are very highly conserved in insects, there's one short wavelength one here that sees in the ultraviolet, one in the blue, and maybe one in the green. And then created this composite curve of the three, and then parameterized it by a whole bunch of field studies that manipulated the content of spectrum to look at attraction levels. And so this curve here is a relative sensitivity showing that the peak attractiveness of insects in general is down here, 350, 400, somewhere in that range, which is way in the violet, the short wavelengths for humans, and extends out into the greens where humans are seeing very, very well, but not as much as down here. And so what we can do is use things like this to try to predict the effects of lights of different spectrum and see how well we're doing. And so CCC there is important, which is the correlated color temperature, but the correlated color temperature is just a summary of what we call the spectral power distribution of the lamp. And so on the one hand, so we know we've got the sensitivity of the insects first, and now here we've got a couple of curves that are showing us the emissions of different lamp types. And this is a particular study that my students and I did a few years ago, looking at a compact fluorescent light, which was had a color temperature of 2700 Kelvin, which is the summary metric, sort of adds it all up together and says, yeah, the light's going to kind of look like something right around there. And then these at the top are all LEDs. One, you can see how different they are. They have different spikes. Two of these are 2700 Kelvin, and one of them is 3000 Kelvin. The 3000 Kelvin is this one here, the purple line. So it has more blue relatively, so it's going to look more sort of neutral white. And the 2700 Kelvin have more emissions over here in the red. And these emissions curves, these spectral power distributions, were custom tuned. We calculated we had a tunable LED where we could create 2700 Kelvin in a number of different ways and 3000 Kelvin in a number of different ways. And we actually moved them around. The experiment was done with Philips Lighting. They provided the tunable LEDs, and we actually sort of messed around with the spectrum to try to avoid the areas that insects were going to be most attracted to while still achieving the same color of light. I show this as sort of an example of the first putting this approach into practice that I was able to do with my students. So you take these spectral power distributions, which we've already done some sort of back engineering on to reduce impacts, and then you put them over a pan that has some water in it. Insects are attracted to the light. They end up in the pan and collected there. And we did this in 3 locations around Los Angeles, 6 sites per location, multiple nights, randomizing the locations, etc. So it was a big experimental setup, and collected something on the order of 5,000 insects, plus having a control for each of these as well, one of those pans with a dummy light but no light illuminated. And then sorted them down to a taxonomic group, and we're able then to show and compare, taking into account the brightness, because there were some differences in brightness of these different lights, but taking them into account, what we found was that the 3000 Kelvin, which is this sort of lighter color here, that's light A, was more attractive than the 2700 Kelvin LEDs over here. The complex fluorescence, it had some spikes down there in the short wavelengths, was in fact, even though it had the same color temperature, more attractive than the other lamps, all of the LEDs, and that the off-the-shelf LED, which was this one here, which was not, in fact, tuned, attracted more of the moths here, the Lepidoptera. And if you add them all up, each of these in their way were significant on the multiple comparisons. But especially here in the Lepidoptera, you can see the decrease in attraction by tuning the light to avoid those areas of the spectrum. And so after doing this study, it really led me to want to look at this more and how we might use spectrum across the board, even if we're using the coarse measure of correlated color temperature, this was kind of proof of concept that we could calculate and minimize attraction and by adjusting the spectral power distribution if we knew what the response curve was from the organism. So that led here. And what here is, these are all different lighting sources where we have these emissions distributions. D65 up here at the top is actually a standard for daylight. And that's defined and internationally agreed upon. And that has the highest color temperature. And you can see I've got the rainbow here on the bottom to show you what areas of the spectrum are in each of these. And then the lowest color temperature down here is low pressure sodium. So low pressure sodium is used very much around observatories and other highly sensitive environmental situations, and basically is a yellow light. High pressure sodium is a orange yellow light and does have some emissions down here in the blue. But all the rest of these include many commercially available LEDs. So for example, the one I have labeled Los Angeles LED, that's sort of a standard 4,000 Kelvin LED. People buy things called yard blasters. I don't even know why, but it's got this power distribution. And then we have a series of LEDs that have reduced emissions in the blue. So PC amber, that's a phosphor-coated amber, a yellow light. You see there's very little blue, but some of it down there. This is a filtered LED that has a filter put on it to reduce the blue emissions. And a number of these, this is also filtered. Anna's light is a filtered LED, etc. So we took this sort of, and then metal halide is also a commonly used outdoor light. So we took all of these spectral power distributions, and then we wanted to compare them against a whole suite of biological responses. Now, the insect attraction curve that I showed you before is basically right here. That one is labeled insect attraction. There's also an old curve for bees. There's one for a few for sea turtles. We included in the study the human melanopic response, which is the suppression of melatonin in humans, the sort of sensitivity of the human body, circadian rhythms. We included the daytime human vision, daytime nighttime vision. And then there's a curve for salmon, moths, and then the one bird one that I was able to get and include in this study was for Newell's shearwater, which wasn't easy to find, and it wasn't from field studies. It was a measurement in the lab using the sensitivity of the eye using electroretinogram, where they hook some electrodes up to it. But it was, and this is actually digitized out of a thesis from the 80s, the 1980s. And so that's the one, but it is interesting as we go along to have that in there and be able to incorporate this seabird into that study. Then photosynthesis here, if we're looking at impacts on plants, and then a number of physical metrics to look at the sky glow, the scattering of light in the sky in different physical ways, and then a composite, the Starlight Index, which just comes out of the sort of astronomical light pollution world. So basically, if you take all of those curves and multiply them by all of those curves, you get something that looks like this. And I don't expect anyone to sort of remember exactly what's on here, but you can go and look at it and download the code and run this yourself. We set this up as a open source tool that you can deploy on your website. And what it has is spectral power distributions of lights going down one side and these biological or physical response curves going on the other. And then we compare everything to an additional lux of daytime light of D65. So you see that the top here is 100%. So that's what we compare everything out. So what it says is, for each additional lux of, say, the Los Angeles LED, how does that compare in terms of affecting moth attraction as if you had had an additional lux of D65 of daytime light? And the answer is it's about 58% effective as daytime light. And then we can start to compare these. So if Los Angeles LEDs are 58% and high pressure sodium here is 37%, I can then predict based on this particular LED spectral power distribution and this spectral power distribution that we should have a decrease of, what would that be? Maybe about a third in insect attraction going from this LED to that high pressure sodium, or the other way around, if we're converting from old high pressure sodium to LED, we can predict an increase in insect attraction. And so we can go about and look at these things across all the different species, the curves that we have. And current research is trying to get more of those curves, dredge them out of the literature so that we can be able to expand the taxonomic breadth of what we're talking about here. So then what we did is we took them and we sort of compressed it down into 4 categories. Insects, so all the insect curves, sea turtles, newel shearwater, the one bird, and the juvenile salmon, and then a wildlife index that really just weighted all 4 of those together. And then the colors represent the color temperature. Even though we understand that color temperature is imperfect, it is still something that we can measure. And you can see that D65 has a very high color temperature, 6,500 Kelvin. Anna's light here is the lowest. It's almost down to 1,000 because it really just chops out all of the blue. Low pressure sodium is down here as well. And then we've ranked them by their summary. And you can see that we've sorted it by the wildlife index, all of them together, to say, if you had to pick one light type that was the best for wildlife, what would it be? And the answer is really low pressure sodium. But unfortunately, low pressure sodium isn't being made anymore. And so LEDs are the future of outdoor lighting. And we have some LEDs down here. This is PC amber. It does quite well. Not as good as low pressure sodium, but very good. And these here are a couple of filtered LEDs. I'll show you an example of one of those. High pressure sodium, turns out, was pretty good that we had for all those years of orange light. It was actually really good from a wildlife perspective. The other thing I want you to notice on this chart is that the order is not the same in all of these subgroups. Juvenile salmon sort of correlates pretty well, but you'll see that the shearwaters, the Anna's light for some reason isn't as good for shearwaters. Anna's light's another one of these filtered LEDs. It's a custom product from a manufacturer that we collaborated with. Whereas low pressure sodium does better. And you can also notice the difference in the highest to the lowest is less for newel shearwater than it is for some other things, both insects, salmon, etc. And that's actually an important point that we'll come back to. We then took it another step and put ourselves in the feet of the lighting engineers and said, what if we also put a penalty in there for lights that have very bad color rendering? And we say that your light has to meet a certain color rendering index because this is what we're told, at least by the lighting engineers and designers, whether they're right or not, but we have to have some good color. People don't like it when they can't see color at night. So if we just add a sort of a threshold for color rendering and reorder them, we find that, and so this sort of penalizes the lights that have the worst color rendering index. So that puts low pressure sodium up here because it's a very low color rendering index. And it puts the phosphor coated amber lights down here because they have that little bit of blue in them that actually lets you see some color at the same time as you have a preponderance of the longer wavelengths. So we can start here at least, and the reason I showed you the website and the code is you can bring your own response curves and you can bring your own spectral power distributions for the lamps. Just have to download the files and update them, and then it'll do all the calculations for you. We then also tested this with, and I tried to correct the spelling on Airam's last name, and I apologize there, with the data from Rodríguez et al. 2017 that we then reanalyzed, and Airam was a co-author on the 2018 paper looking at predicting attraction of short-term shearwaters to 3 different light sources, where they had a high pressure sodium, an LED, and a metal halide. And so the things here that we want to look at is actinic power per lux. This is using our method of trying to use, and we use the Newell Shearwater visual response curve, which is probably wrong, but it's closer than anything else that we have at the moment. So it's like the closest species we had to it, and predicted this levels of attraction. And so what we predict there is that the high pressure sodium would have the least, and the LED, and then the metal halide. And then we had brightness and color temperature. And you can see that the color temperature was quite similar between the LED and the metal halide, but our approach using actinic power, the action spectrum times the spectral power distribution, sort of sorted them out. And when you compare all four of these models, this model actually has the greatest explanatory power, significantly explanatory power. So this was sort of our back test about whether this approach worked, of spectral power distribution times action spectrum or response of the organism, leading to a prediction of the impacts. And then, so that was published, and get a lot of questions then from different folks in the lighting industry about other metrics beyond correlated color temperature, because there's this discomfort about correlated color temperature, because there are ways that you can kind of game the system and make something look two lights can look very different and have the same correlated color temperature. So we ran these regressions looking at correlated color temperature and the percent of light emitted in the blue, defined as under 530 nanometers, the melanopic response curve, which is the human melanopic response, and compared them across these different averages. And the two things that I want you to see here again, one is that the R-squareds, human melanopic response is quite good, but correlated color temperature is also pretty good. But it only works for the sets of lights that don't have violet or ultraviolet. And I have to add that caveat here. The human melanopic response curve as an indicator of all other biological responses only works when you don't have violet or ultraviolet, because it is not sensitive down into the ultraviolet, and insects, for example, are. And then the other thing to notice is sort of how much you can affect these sort of responses based on actual real world lights. And the thing I wanted to point out here is that the Newell Shearwater, these curves are all quite high up on the scale, and they're all pretty shallow. There's a reasonable R-squared on here, but what it indicates is that we cannot, unlike say for insects, go from 80% attraction compared to daylight down to 25% attraction or 20% attraction. We can maybe go from 85 to 65, right? There's a much narrower range using lights there, and again, all the points on here are sort of real, actual lights that you could use. I mean, kerosene oil's in there because we wanted to compare with old lighthouse lights and things like that. But there's much less gain to be made around spectrum looking at shearwaters than there is for sea turtles, and spectrum is a well-known mitigation for sea turtles or insects or salmon. And that's something that I just wanted to point out. Not that it's not impossible, not that it doesn't have some explanatory value, but what it means is the first 4 points of your mitigation approaches, in terms of intensity and direction and duration, and need, are the ones that you have to rely on in addition to any strategy that involves spectrum when it comes to seabirds. And now there's good reasons to want to keep it at low color temperature also for astronomical light pollution and the scattering in the atmosphere. But strictly from a seabird perspective, we've got some gains to make, and you can certainly make it worse, but it has to be part of a comprehensive strategy. And so this is putting them all together, that average wildlife sensitivity, just so that you can see them summarized again here. The lowest impact here, low pressure sodium, all compared to this baseline of what if you had daylight. The full moon actually isn't exactly like daylight. It's a bit redshifted, so an additional lux of full moon doesn't behave the same way as an additional lux of daylight would have. And then we can see these LEDs in here. This is a 4200 Kelvin LED, a 3000 Kelvin LED. And then the LEDs that are down here in the lower impact areas, these are filtered LEDs. This telescope light's a filtered light, filter, high pressure sodium, low pressure sodium, phosphor coated amber, filtered light. And so those are our options if we want to take a comprehensive approach, given what we know right now. And I'll say that, always can caveat, given what we know right now, this is an approach that we can take to minimize impacts of things like this. And so I kind of wanted to end here the presentation portion of our time together with some images. So an unshielded light that is quite bright causes huge contrast. This is lights over water. You can see the difference between high pressure sodium and full spectrum lights. That's actually going to make a difference. There's salmon in here. This is a Boeing facility up in Washington State in the United States. All that light going down into the water. This would also be bad if they had seabirds trying to get down to the ocean. This would be bad as well. So these are not shielded, they're not directional. The new ones are certainly not taking into account anything other than the desire for human observation and human vision. Even down to the littlest thing, these little garden LED lights that are very blue because they're energy efficient, they're older technology that can affect, and we've got studies now showing that'll affect the distribution of your slugs in your garden, of your beetles in your garden. So it goes all the way from the area lighting down to the smallest yard light. Here's a street light, completely high pressure sodium, so the spectrum is good, but completely unshielded. So we want to deal with these issues. So some good designs here of people trying to balance these needs, and I put the designers on here because they deserve credit for being willing to go here. Here we're using a low color temperature light. It's low, it's keeping the light low. Here's another, even though we have a little bit of area light here, it's incredibly shielded, very directional, trying to keep the light where it needs to go. Here's another one in another context of this low lamp lighting. And then we can extend this also to whole street lighting systems and whole villages and whatnot. And this has been pioneered first using color around observatories. It has a long, multiple decadal history. But this is using LEDs, filtered LEDs in this instance. This is an original photograph of a streetlight system that has been converted out to filtered LEDs. And there's probably still some gains to be made with shielding and intensity and whatnot. But the analysis here, and this comes from Chips and Wafers LED company in Arizona. This is the amount of red in that picture, the amount of green, and the blue. You can see there's just a few white lights out there. You can see them show up on the blue channel, and then the moon up here at the top, obviously, with a lot of blue in it. So as a means, the technology is out there to do that. But in the instance of seabird attraction and bird attraction, there's probably less there's gains to be made in spectrum, and there's every reason from a comprehensive environmental perspective to go with lower color temperatures, more yellow and red. But every time we use that strategy, we need to also be talking about and being sure that we address the directionality of the lights, the timing of the lights. Can they be turned off? Can they be dimmed? Can they be, have a seasonal turn off when there's particularly sensitive things going on, and really lighting only where that is necessary. So that's how I've gotten to this point of talking about spectrum as a mitigation approach, and tempering it with the reality of this always needing to be a comprehensive, multi-pronged strategy to minimize light pollution and the adverse consequences to birds, and also, of course, to people, both because people enjoy birds and because people are adversely affected by extending their nighttime activity too much as well, which we all, I hope, have an intuitive awareness of. So that's what I wanted to share with you here this morning. I want to have some back and forth questions, and also you can, if we don't end up talking, you can email me, or you can contact me on Twitter or tag me there, and I'll engage to discuss these issues further. And I really look forward to our conversations as we go forward. And thank you so much to the organizing committee for inviting me to this really interesting and exciting event today.
Great presentation. I really like it. And well, I really enjoyed, especially the part of the newly share waters.
Yeah.
So, and like you said, well, I just would like that the people, participating in the webinar, ask you some questions if they have doubts, and if they want to learn more, well, and actually enjoy the opportunity to actually talk to you and learn more about the impact of the light pollution. Anyone, any questions?
If you want to ask a question, you can go ahead.
Yeah, I was really interested in the part of the spectrum with the newly share waters.
Yeah.
Because you really said that they do not have actually a huge margin like the other species in terms of spectrum.
Yeah.
So that is really sad. So,
yeah, I know. Yeah, and the thing.
Do you think that is for all the seabirds, or?
Okay, so there's this dissertation by Reed in 1982.
Oh, yeah.
Okay, the Reed 82, and I can send you a copy if you want it. And he has a series of curves of different seabirds, and they're all pretty similar. And the problem is that now that's just an electroretinogram, so I can't say that that's 100% behavior.
Yeah.
Right? So that's where you need the tests. It's like, okay, we think the vision is going this way. And then, of course, there's different oil droplets and all this sort of stuff, and different species. And there's a grad student in Hawaii now named Hana Moon, and she's working on this for her dissertation, like this measurement issue and spectral responses and whatnot for seabirds there. Shearwaters, I think it's Newell's she's working on.
Yeah.
And so I always want to have this word of caution that yes, we should care about spectrum. Yes, we should design, and there's many, many reasons to design with it, but it's not going to fix everything.
Exactly.
Because the industry can look at it and say, well, it's kind of my get out of jail free card. Oh, if it's this color, it can be as bright as I want it. Well, no, there's a, even if you have a tiny bit of blue, if it's super bright, it's still bright,
Yeah, that is true. Well, luckily, the guidelines that we are using, it's mostly the directing, having the shield, the decreasing the intensity, do not have the glass, so do not disperse so much, all this kind of stuff, and the color, of course. But we are not getting the ones that we should, or at least the better ones, because we are using only neutral ones. Because the company said that the PC amber and the yellow ones have lower longevity, so and that is for them in terms of economics. It's and we have to get to an optimal. That was the issue, and we just try to use the neutral ones, but the LED 3000, but that they on the fledgling periods they decrease, and they have directed lights, shielding, all this kind of stuff. Well, it was the possible, at least for now.
And so when people are worried about longevity, the last picture that I showed you, the yellow lights,
Yeah.
those are actually a 3000 Kelvin driver
Yeah.
with a filter. So
Yeah.
and I didn't want to put the full name or the website of the company in it in the talk, but
Okay.
there is a company that does these filters, and they're doing this in Hawaii for exactly this purpose. So they're using, and I have a bunch, they send me some to play around with. And so you can take it as a warm white, and so you can also do it with the 2700 driver.
Yeah.
So a
Yeah.
2700 driver with a filter, and there's only like a 5% loss of effectiveness by having the filter on it,
Oh, so it's not much.
which is
Yeah.
not much at all. So you've
Yeah,
got the
yeah.
longevity of the 2700 Kelvin that is now, or should be soon, standard,
Yeah.
and then
Yeah.
a filter on top of it, and the directionality.
Yeah, I think it will work. We are going to monitor, and we have a researcher now. She's doing the PhD that she's working with the core waters in terms of what you are doing with the spectrum, trying to see how they actually the vision, how it works for
Yeah,
the fledglings.
good.
So
Good.
that is actually good for us on the Azores. And
Yep.
well, thank you very much for the
Yeah,
for
it's
the
fun.
enlightening. It was
Okay.
a pleasure. And now it's going to be much more interesting when I'm reading the articles, because I know the face.
Yeah, exactly.
Bye.
Thank you.
Thank you.
All right, take care.
Bem, continuamos com a próxima apresentação. O próximo orador é o Airam. Olá, Airam, ¿qué tal?
Hola, muy bien. ¿Qué tal por ahí?
También muy bien, gracias. Bueno, voy a seguir en portugués, que es mejor para mí. O Airam é licenciado em Biologia pela Universidade de La Laguna em 2005 e completou os seus estudos de doutoramento na Universidade de Sevilha em 2012. Tem interesse em aves marinhas e também numa das fontes de mortalidade que o marcou quando era apenas uma criança. Como canário, foi testemunha do encadeamento de centenas de pardelas como consequência da poluição luminosa, e este interesse científico levou a visitar várias localidades onde se registavam interações entre as luzes e as aves marinhas, como o Havaí, a Austrália, a Ilha da Reunião e as Ilhas Baleares. Dirigiu o projeto Ecolights for Seabirds, financiado pelas ações Marie Curie da União Europeia, e atualmente colabora também no projeto Luminaves, liderando um projeto sobre a poluição luminosa nas Ilhas Baleares, financiado pelo Agreement for the Conservation of Albatrosses and Petrels. Já publicou mais de 15 estudos sobre o impacto da poluição luminosa em aves marinhas e hoje falará um pouco sobre o que é que sabemos e o que é que deveríamos saber para mitigar esta fonte de mortalidade. Além disso, também apresentará alguns resultados do projeto Luminaves e tentará preencher alguma desta falta de informação. Quando queiras, Airam, podes começar.
Muchas gracias, Katia, por esta introducción. Voy a intentar compartir la pantalla, a ver si me acuerdo. Compartir esta pantalla. Ok. Aquí estoy yo. Ok. Bueno, pues mi presentación, la presentación de hoy va acerca de qué sabemos acerca del impacto de la contaminación lumínica en las aves marinas, qué deberíamos saber y en qué aspectos del proyecto Luminaves está ayudando a saber de este problema. ¿Vale? Antes de empezar, me gustaría hablar un poco de las aves marinas, y en concreto de las pardelas y de los peteles. Pardelas y peteles son aves marinas, como su propio nombre indica, pasan la mayor parte de su vida en el mar. Pasan tanto tiempo en el mar que solo vienen a tierra solo para nidificar. De hecho, si pudieran poner su único huevo en el mar, si tuvieran un sustrato sólido en el mar, no vendrían a tierra. En el mar pescan y pasan la mayor parte de su vida. Pueden pasar hasta 3, 4, 5, 6, 7 años sin tocar tierra, que es la edad a la que los jóvenes vuelven a las colonias guía para reproducirse. Y cuando vuelven a las colonias a reproducirse, esas visitas las suelen hacer durante la noche. Además, para nidificar suelen utilizar lugares muy inaccesibles, como islas, roques, acantilados o lugares de muy difícil acceso. Una vez llegan a estos lugares, nidifican bajo tierra y ponen un único huevo, el cual solo produce un único pollo, por supuesto, en cada uno de los intentos de cría que se produce una al año. ¿Vale? Después, durante el día, se van a alimentarse al mar y vuelven otra vez durante la noche. Este grupo de aves marinas es también uno de los grupos de aves más amenazados del mundo. Según la IUCN, más del 50% de las especies están amenazadas. Y entre las amenazas que hay de estas especies, las más importantes son la introducción de depredadores marinos, de depredadores mamíferos, depredadores introducidos principalmente para candidatos, también las interacciones con las pesquerías industriales, la caza ilegal, también la contaminación marina, principalmente por hidrocarburos, incluso por plástico, la destrucción del hábitat de nidificación, como puede ser el caso de construcciones humanas, y también la alteración del hábitat de cría a través de la contaminación lumínica. Y este es de lo que vamos a hablar. El principal problema que tienen este grupo de aves marinas es el siguiente. Los pollos, cuando están en el nido, llega un momento en que deben emanciparse. Y ese primer vuelo lo hacen por la noche. Ese primer vuelo vuelan desde las colonias de cría hacia el mar. Y en el camino, en ese vuelo nocturno, se encuentran con las luces de nuestras ciudades, nuestros pueblos, estadios deportivos, zonas industriales, etc. Eso les produce que se deslumbren y que se traigan y que acaben cayendo en las carreteras, bueno, en la superficie en cuestión, en estas áreas. Y después queden vulnerables a otras fuentes de mortalidad. Si consiguen sobrevivir a ese primer impacto, luego quedan susceptibles a otras fuentes de mortalidad, como pueden ser atropellos por coches, predadores introducidos como gatos o perros, o incluso otras especies, como pueden ser lechuzas introducidas, o que quedar atrapadas en trampas, tipo alcantarillas, o tubos de drenaje, o zonas donde nadie los puede recoger, como una terraza, lugares donde no pueden salir y por tanto acaban muriendo de hambre o inanición. También pueden caer en charcos de aceite, pintura o cualquier otro líquido peligroso para su plumaje. Y esto causa entonces una gran mortalidad concentrada justamente en el periodo de emancipación de los pollos. Este es un fenómeno a escala mundial, pasa prácticamente en todos los archipiélagos del mundo donde coexisten humanos y aves marinas, aves marinas, y no solo está ligado a las aves, a pardelas y petreles, también hay otras especies como los patos marinos, los eiders o los frailecillos, que también son susceptibles de este tipo de mortalidad. Los puntos que les hemos mostrado en este mapa no son todos los puntos que ocurren, faltan muchos más, pero es simplemente para dar una idea de cuánto de amplio es el fenómeno en el mundo. Pero la pregunta del millón es ¿por qué son forzados a aterrizar? O sea, ¿por qué son atraídos por las luces? Pues la respuesta no está muy clara, pero al menos se han propuesto tres hipótesis. La primera de ellas es que se propone que confunden la iluminación artificial, las luces nocturnas, con presas bioluminiscentes. Hay que tener en cuenta que estos individuos son ejemplares jóvenes que no han visto nunca una presa bioluminiscente en la naturaleza. Ellos se alimentan principalmente de la comida regurgitada y semidigerida que le traen los padres. Por tanto, en realidad nunca han visto un calamar en el mar. Y podrían tener de forma innata esa atracción hacia la luz para ir a alimentarse. Aquí les he traído dos fotos. En la parte izquierda, una foto de una ciudad tomada desde una parte elevada, y a la parte derecha pueden ver un calamar bioluminiscente. Si eres un individuo inexperto y joven, que nunca has nadado en el mar, ni conoces muy bien tus presas, podrías llegar a confundirte con ellas. La segunda hipótesis es la pérdida de los cielos nocturnos naturales, es decir, las señales de los cielos nocturnos naturales. Por ejemplo, la luna, la luz de las estrellas, o incluso de la Vía Láctea, o incluso el reflejo del cielo nocturno en el mar. Aquí les he traído una foto de un puente donde se ve la luna llena, que justamente podría parecer coincidir con una luz de las del puente, y como su reflejo en el mar es bastante parecido. Y la tercera hipótesis que se ha propuesto es acerca del desarrollo visual del sistema de los pollos. Se sabe que estos pollos cuando se emancipan, es decir, cuando dejan su nido y se van hacia el mar, su sistema visual no está completamente desarrollado. Y esto podría tener algo que ver. Pero para mitigar la mortalidad producida por la contaminación lumínica, la principal acción que se lleva a cabo en muchos archipiélagos del mundo son las campañas de rescate. Estas campañas de rescate son normalmente coordinadas por los gobiernos locales, por ONG o por grupos de voluntarios que se organizan para pedirle a la gente que si encuentran algún ave en el suelo, pues que lo rescaten y que lo lleven a lugares a estaciones de rescate, o sea, lugares donde después las campañas van a pasar por allí para el personal cualificado va a pasar por allí para recoger las aves, examinarlas, y si están bien, entonces le damos una segunda oportunidad a ese ave liberándolas en un lugar seguro, sea en el mar o otra vez en las colonias. Esta medida de mitigación, la contaminación lumínica, es la principal medida que se lleva a cabo en todo el mundo. Pero hay que tener en cuenta que esta medida es una medida paliativa. Paliativa en el sentido de que solo rescata aves que ya han sido anexadas, que ya han sido, por tanto, digamos que no es la solución, es solo un parche. Pero bueno, a pesar de que sea solo un parche, hay que decir que estas campañas de rescate son bastante útiles, puesto que, por un lado, permiten minimizar la mortalidad, lo cual es lo más importante, y segundo, permiten proporcionar mucha información acerca de las aves marinas y de este proceso de la atracción hacia la luz. Y la mayor parte de la información que tenemos de este fenómeno viene de las campañas de rescate. Y ahora vamos a ver qué información nos ha dado estas campañas de rescate. Por ejemplo, sabemos que hay un claro efecto del ciclo lunar en el número de aves amerizadas. Durante las noches de luna llena, el número de individuos que son atraídos es mucho menor que en nuestra luna nueva. Aquí les traigo en estos 4 gráficos, les traigo 4 histogramas en el que se muestra en el eje de las X el número de aves rescatados por cada noche y en el eje de las Y el número de aves. Como podemos ver, hay 3 años, en el año de 1998, en el año 2003 y en el año 2006, donde la luna llena, que está representada en estos gráficos por esa gran barra gris que incluye más o menos 2 días de la luna llena, podemos ver como el número de pollos durante esas barras decreció significativamente. Hay que tener en cuenta que ese periodo, en torno al 4 o 5 de noviembre, es el periodo de la emancipación media de los pollos, es decir, la mayor parte, cuando acumula muchos años de datos, la fecha media del pico máximo de emancipación de los jóvenes está entre el 4 y el 5 de noviembre. En el año 2005 podemos ver cómo las lunas llenas no interaccionaron con esa fecha, y si se fijan podemos ver cómo en el eje de las X, en la escala, es mucho mayor. En una única noche cayeron más de 150 pollos, comparado a los 60 o 80 que caían en los años, en los otros 3 años, ¿no? Así pues, aquellos años en los que la luna llena coincide con la fecha media de los pollos, el número de aves que se rescatan es mucho menor. Y esto ha sido descrito en Hawái como una especie que es la pardela de Niue. En este caso, la pardela se emancipa un poquito antes, en el mes de octubre, y en torno a mediados del mes de octubre. Como podemos ver en esta gráfica, aquellos años en que la luna llena coincide con ese 15 de octubre, el número de aves es bastante inferior. ¿Vale? En esta gráfica se representan, cada punto representa un año. Y en el eje de las Y se representa el número de aves que se rescatan cada año. Pero aparte de eso, las campañas de rescate también han dado información acerca de estas especies que son tan raras y tan esquivas. Hay que tener en cuenta que para muchas de estas especies no se conocen ni las áreas de cría, donde crían. ¿Vale? Y de hecho, aquí les traigo una cita del profesor Michael Grupo, de su famoso libro Albatros and Petrels Across the World, en el que él mismo dice que es remarcable que por muchos años nuestra mejor evidencia de la supervivencia continuada de una especie de petrel viene de aves aterrizadas a las luces, atraídas a las luces. Bueno, en el caso de Canarias, utilizamos precisamente el número de aves rescatadas durante las campañas, desde el año 1990 hasta el año 2010, para evaluar las tendencias poblacionales de las especies. Esto lo hicimos para 3 especies, para la pardela cenicienta, arriba, el petrel de Bulber en medio y la pardela chica. Como pueden ver, las tendencias son bastante diferentes. Mientras que la pardela cenicienta incrementa a lo largo de los años el número de pollos, de hecho, esto es hasta el 2010, si miramos los años de hasta 2020, el incremento seguiría incrementándose. Esa tendencia de la pardela cenicienta es diferente a las tendencias del petrel de Bulber y de la pardela chica, los cuales muestran una tendencia más o menos estable, mientras que la pardela chica muestra un claro declive. Estos datos hay que tenerlos en cuenta relativos a la contaminación lumínica que ha habido en Canarias durante este periodo, que claramente ha incrementado, y también a la concienciación de los ciudadanos durante este periodo, que posiblemente, y es esperable, todo parece que sí, que la concienciación haya también aumentado. Entonces, en este contexto, en el que la contaminación lumínica aumenta y la concienciación también aumenta, sería esperable que cada vez se recogieran más aves hasta llegar a un límite, a un tope máximo. Las dos tendencias del petrel de Bulber y de la pardela chica claramente indican que sus poblaciones están declinando. Y esto es muy importante. Aunque parezca que es una información muy básica, hay que tener en cuenta que, por ejemplo, para estas dos especies, la mejor evidencia que tenemos de sus tendencias poblacionales son esta información que les he presentado. Así que evalúen si no es importante las campañas de rescate. Pero las campañas de rescate también han informado acerca de la condición de las aves aterrizadas. La condición principalmente medida como el peso. Para 4 especies de aves marinas se ha visto que la condición de los individuos decrece a lo largo de la campaña de rescate. Es decir, los individuos que se rescatan al principio están más gordos que los individuos que se rescatan al final. Y alguien podría decir, bueno, eso es normal, porque los padres abandonan a los pollos en sus nidos y por tanto los pollos al no comer, pues no pueden engordar y lo único que hacen es crecer el peso. Y es verdad, es así. Pero también es verdad que este es un rasgo muy importante para estas aves marinas. El peso, en el momento de emanciparse, el peso cuando el pollo se emancipa, es una medida muy importante que predice la capacidad que tienen los individuos de sobrevivir los meses posteriores en el mar y posteriormente reclutar en la población reproductora. Es decir, los pájaros más gordos tienen mayor probabilidad de sobrevivir y de llegar a adultos y de criar en la población. ¿Vale? Por tanto, se tiene un claro mensaje. Es mejor rescatar al principio de la campaña que al final de la campaña, cuando los individuos no tienen ya prácticamente ninguna probabilidad, o tienen menores probabilidades de sobrevivir. Puestos a priorizar, priorizaríamos el principio de la campaña. Pero claro, esta comparación solo se ha hecho con aves que han sido aterrizadas, o sea, que han sido afectadas por la luz. Alguien podría pensar, bueno, pero estas aves que han sido afectadas por la luz son iguales a los otros pollos que no se afectan por la luz. O sea, ¿hay algún tipo de hándicap de los individuos susceptibles de ser aterrizados por las luces? Bueno, para estudiar esto, lo estudiamos en la isla de Phillip Island, en Australia, con la pardela de Tasmania. Y lo que hicimos allí fue rescatar aves en las carreteras, que habían sido aterrizadas por las luces de la carretera. También nos fuimos a las playas a recoger y a rescatar las pardelas que habían sido orilladas por el oleaje del mar. Y además fuimos a las colonias de cría para mostrar los pesos de los individuos que estaban allí en las colonias de cría. Y eso lo hicimos cada noche durante todo el periodo de emancipación del joven. Y lo que encontramos fue esto. Si nos fijamos en el año de 2015, en la parte de abajo, podemos ver cómo en el caso de las playas, las aves que se rescataban en las playas mostraban un peso muy inferior al de las aves rescatadas en la colonia o en la carretera. En estas gráficas lo que se muestra son box plots, cajas de distribución de la información, y en gris se muestra, esta barra horizontal gris, se muestran los rangos de peso de las aves adultas. Como podemos ver, en el caso de la carretera, hay más de un 50% de las aves rescatadas que se encuentran en ese rango, en el rango del peso de los adultos. Lo cual hace pensar que estas aves tienen una cierta capacidad o cierta probabilidad de sobrevivir y de dar. Y que por tanto las campañas de rescate son útiles y que merecen seguir que se siguen llevando a cabo. De hecho, esto ha sido parcialmente demostrado por un estudio reciente del equipo de Hawái, de Andrés Raine, con la parcela de Newell. Ellos lo que hicieron fue marcar aves con GPS y estudiar la migración y la supervivencia de aves que habían sido rescatadas y de aves naturales en los nidos. O sea, que no habían sido afectadas por las luces. Lo que vieron fue que, aunque la supervivencia parece ser ligeramente inferior en las aves que han sido rescatadas y afectadas por la contaminación lumínica, muchas de ellas tienen una supervivencia similar a las naturales y, por tanto, las campañas de rescate son meritorias. Son merecedoras de seguirse llevando a cabo. Pero, ¿y qué sabemos de los tipos de luz? ¿Son todos los tipos de luz susceptibles de atraer aves? Pues para estudiar esto lo que hicimos fue un experimento también en la isla de Phillip Island con la parcela de Tasmania, donde testamos 3 tipos de luces: LED blancos, metales halogenados y sodio a alta presión, ¿vale? 3 tipos de luces que randomizamos cada noche durante la época reproductora, de la época de la emancipación de los pollos. Lo que vimos fue que el número de aves amerizadas fue mucho mayor en el caso de los metales halogenados que en el caso de las luces de sodio a alta presión. Y el caso de los LED blancos fueron ligeramente superiores, aunque no significativamente, al caso de las luces de sodio a alta presión. Hay que tener en cuenta que aquí no solo testábamos el espectro lumínico de las fuentes de luz, sino también el tipo de lámpara. Nosotros usamos 3 tipos de lámparas. 3 tipos de lámparas estaban disponibles para la compra de la gente y estábamos interesados en hacer un trabajo que fuera realmente aplicado a los usuarios, ¿no? Es decir, la gente puede comprar diferentes tipos de luces, pues de las luces que había en el mercado, pues usamos este tipo de luz. En cuanto a la condición corporal, no encontramos ninguna diferencia entre los tipos de luces, o sea, por ese lado, bien. Otra pregunta importante que ha ayudado a responder las campañas de rescate es dónde caen, la distribución espacial. Por ejemplo, esta es la isla de Tenerife y los números que se muestran ahí son los porcentajes de aves rescatadas en cada uno de los municipios. En Tenerife, gracias a las campañas de rescate, se sabe que aproximadamente el 75% de las aves se rescatan en el suroeste de la isla. Y concretamente, aquí, en la parte turística de los que quedan en las Américas, en el sur de Tenerife, se rescatan más del 50% de las aves. Esto es muy interesante para las campañas, pero desde el punto de vista conservacionista, sería muy interesante saber de dónde vienen. Es decir, esos individuos que se rescatan en el sur, que son más del 50%, vienen de colonias cercanas o vienen de colonias lejanas de los núcleos más importantes, como pueda ser el macizo de Plymouth o el macizo de Marinas. Esto es una cuestión muy importante de cara a la conservación, puesto que implica que toda la población insular está afectada por las luces o solo una parte de la población insular está afectada por las luces. Para intentar responder a esta pregunta, lo que hicimos fue, nos fuimos a las colonias, intentamos capturar a los pollos en sus nidos, marcamos esas aves con un GPS, con un dispositivo GPS en la espalda, en los talones, liberamos otra vez esos individuos en sus nidos, nos metimos bajo tierra en sus nidos, y esperamos a que esas aves salieran hacia el mar. Mientras, hicimos una pequeña campaña de rescate, una pequeña campaña de sensibilización para que la gente supiera que si encontraba una ave marcada que tenía que llamarnos o ponerse en contacto con la coordinación de la campaña de rescate, con el Cabildo de Tenerife. Y bueno, y después esperamos a que el Cabildo de Tenerife se pusiera en contacto con nosotros para decirnos, oye, tenemos un pájaro marcado, y íbamos, recogíamos el GPS y descargábamos la información. Lo que obtuvimos fue lo siguiente. Esta es otra vez la isla de Tenerife. En azul les muestro las áreas contaminadas. Aquí Santa Cruz de Tenerife, que es la capital, y aquí las zonas del sur de Tenerife, las zonas turísticas de los ecosistemas de las Américas, de la zona de. Los puntos negros indican colonias donde llevamos a cabo marcajes, anillamientos, marcajes, ¿vale? Y las líneas rojas indican los vuelos de los individuos desde sus colonias de cría, desde donde fueron anillados, hasta los lugares de aterrizaje. Las líneas discontinuas son recuperaciones de anillas, es decir, no sabemos por dónde el ave voló, pero sabemos que se anilló en tal sitio y se recuperó en tal otro sitio, ¿vale? Lo importante de aquí es que el 50% de las aves fueron rescatadas a menos de 3 kilómetros de su nido. Es decir, el 50% de los pollos cayó muy cerca de su nido. La distancia máxima fueron 16 kilómetros de individuos que se liberaron aquí, en la zona de Plymouth, y se acabaron en Santa Cruz, ¿vale? Y además hay otro aspecto muy importante, que las colonias que están tierra adentro, es decir, estas que están aquí, estas que están aquí, o estas que están aquí, tienen mayor probabilidad a ser afectadas por la contaminación lumínica que las colonias que están justo en el mar. ¿Por qué? Porque las colonias que están justo en el mar, los individuos simplemente se dejan caer y ya están en el mar. En el caso de las colonias que están tierra adentro, necesitan sobrevolar las zonas expuestas a la contaminación lumínica. Y para ilustrar esto, les traigo aquí un vídeo de una panadera. El ave está aquí, la ven moviéndose, desde ese lugar se libera y parece que se va hacia el mar. Ahí parece que ya está salvada, pero el individuo, por alguna razón, decide volver otra vez a tierra y ahora empieza a volar sobre las zonas urbanizadas, que son esta de ahí y esta de aquí, donde va a llegar, es la zona de Candelaria y el polígono de Weimar, y donde da varias vueltas. El vuelo tiene una duración aproximada de 48 horas y ya está ahí. Ese es el lugar de aterrizaje. Y una longitud de casi 30 kilómetros. Pero bueno, como había dicho, las colonias que estaban tierra adentro son más susceptibles de ser afectadas por la contaminación. Y entonces la pregunta del millón, otra pregunta del millón es, ¿cómo lo hacen los adultos? Aquí en esta diapositiva les traigo dos imágenes de dos colonias que están tierra adentro. En esta se ve el mar a fondo, incluso se ve un barco iluminado en el mar. Las aves tienen que sobrevolar esta zona iluminada para alcanzar la oscuridad de la colonia. Y en la foto recortada que se ve aquí, pues, más de lo mismo. Se ve el mar y cómo hay un cinturón costero de urbe humana iluminada y las aves tienen que llegar a tierra. Bueno, estas colonias, como pueden imaginar, los adultos de estas colonias deben cruzar varias veces durante la reproducción sobre esas zonas iluminadas. Recuerden que estos vuelos se hacen siempre durante la noche. Y claro, los adultos tienen que o turnarse con su pareja para comer los huevos o traer alimento a los pollos. Y esto lo hacen durante prácticamente 6 meses. Para intentar responder a esta pregunta, lo que hicimos fue, otra vez, ir a seleccionar dos colonias al sur de Tenerife, próximas a la zona más turística, la zona más caliente de la contaminación lumínica. Dos colonias que estaban a cierta altitud, a 450 metros y 600 metros, capturar a las aves adultas, colocarles un GPS y hacer el seguimiento de los vuelos que hacían a la llegada y a la salida. Lo que pudimos ver es que cuando ellos dejaban las colonias, es decir, cuando se iban desde las colonias hacia el mar, los vuelos eran bastante directos y bastante rápidos. Parece, a jugar por estos tracks que les muestro aquí, que no hay ningún tipo de interacción con la contaminación lumínica. Es decir, los individuos iban hacia el mar y parece que volaban por encima de la ciudad sin ningún problema. Pero la cosa es bastante diferente cuando los individuos tienen que subir a las colonias. Es decir, cuando van desde el mar hacia las colonias cría. En este caso, los vuelos son mucho más largos y más tortuosos. Los pueden ver aquí como más retorcidos. Además, parece que ellos evitan las luces. Y la pregunta es ¿por qué? Pues la explicación que nosotros le damos, no está todavía en preparación en este artículo, creemos que es porque los individuos tienen que subir desde los 0 metros, desde el nivel del mar, hasta las colonias de cría, que están bastante altas, a 600 metros. Esto, claro, les cuesta un trabajo y las aves tienen que subir esa altura. Eso podría hacer que los individuos vayan más lentos que cuando se dejan caer desde la colonia del mar. Y por tanto, al ir más lento, puede ser que la sensación de predación, de posible riesgo de predación, pueda aumentar. Y por tanto intenten ir por zonas más oscuras. Como digo, esto es solo una hipótesis. Y para que vean dos vuelos de este tipo, les he traído dos viajes, van a ser muy rápidos. El individuo está aquí en el mar y va a subir hacia la colonia de cría que está alrededor de esta zona. ¿Vale? Esta zona de aquí es la zona turística del sur de Tenerife, de las Américas, y miren lo que hace el individuo. Parece que va hacia la luz, pero no, se da vueltas y sube por una zona que está menos urbanizada, se sube a la ladera de un parque natural e intenta evitar toda esta zona de aquí. Y ahora les voy a mostrar otro vídeo, este va a ser mucho más rápido, así que donde se muestra el track de ese individuo que ya acabamos de ver, es este. Aquí la imagen está un poquito a menor escala, se ve ahora todo lo que es los sistemas de América. Y vamos a ver otro individuo que va a subir muy rápido. Fíjense cómo este individuo hace un rodeo y rodea todo lo que es la zona turística, la más urbanizada y la más contaminada, y cómo da un rodeo para alcanzar su colonia cuando de aquí a aquí podía haber ido en línea recta, de una forma mucho más grande. Bueno, y hasta aquí más o menos es esto todo lo que sabemos de los problemas de la contaminación lumínica y las aves marinas. ¿Qué deberíamos saber? Pues bueno, en el año 2017 hicimos un estudio con más de 27 autores de todo el mundo, un estudio en el que revisábamos qué sabíamos y qué deberíamos saber. Y las principales conclusiones que llegamos fueron a estas 5 que están aquí, que aquí las he presentado en castellano, porque meterlas en castellano, pero para los angloparlantes las tienen resumidas en el artículo. Pues estos 5 puntos, el primero de ellos era saber un poco más de la estimación de la mortalidad y los efectos sobre la contaminación sobre la población. Esto ya, como hemos dicho, es bastante difícil debido a la inaccesibilidad de las colonias y también al comportamiento activo de las especies. En el punto número 2, creíamos importante que la determinación de los umbrales y niveles de luz y las distancias seguras era importante, saber a cuánta luz podían ser susceptibles los individuos. También saber algo más del destino de las aves rescatadas, que las aves se rescataban en las campañas y se liberaban. Ahora, gracias a este estudio de los hawaianos, sabemos un poquito más. También el punto número 4, mejorar las campañas de rescate, particularmente en el cuidado veterinario y cómo se deben hacer las liberaciones. Y también, para finalizar, en el punto 5, solicitábamos más investigación acerca de las longitudes de onda y las características de la luz que afectan a las aves. Con esto podríamos diseñar o proponer sistemas de iluminación más amigables para ellas. Pues bueno, el proyecto Luminaves trata prácticamente de estos 5 puntos, pero yo les voy a hablar de al menos 3 de ellos. Del punto número 1, del punto número 2 y del punto número 5. ¿Vale? Para estimar el impacto de la contaminación lumínica en la población, es decir, la proporción de pollos afectados por la contaminación lumínica, debemos saber el número de aves que se rescatan en las campañas, que eso más o menos lo sabemos, pero aparte debemos saber el ciclo reproductor y el número de parejas, para conocer el número de pollos que se produce anualmente por la población. Ese ratio nos daría el impacto de la población. Dentro del marco del proyecto Luminaves, para intentar estimar el éxito reproductor, lo que hemos hecho es seleccionar 117 nidos repartidos por toda la isla de Tenerife, los pueden ver aquí en los puntitos verdes, con la idea de tener una tasa de éxito reproductor lo más homogénea y representativa de la isla, no solo de una colonia. Fíjense que hay muchos nidos que están agrupados en colonias y por tanto el efecto de posibles predadores o de luces o de otras amenazas podría podría sesgar esta estima. Por tanto, nosotros lo que queríamos era tener una estima global para toda la isla. Los nidos se visitaron 3 veces durante la época reproductora y se consiguieron información de 711 nidos en los 3 años. El éxito reproductor promedio fue de 63%. Esto, como decía, era muy importante para estimar el impacto ambiental. Pero eso ya por un lado lo tenemos. Y por otro lado, habría que estimar el tamaño de la población reproductora. Y esto es bastante difícil en estas aves, teniendo en cuenta que crían bajo tierra, visitan las colonias de noche y además una misma entrada a un nido puede albergar varios nidos. Para intentar estimar el tamaño de la población, lo que hicimos fue generar 500 cuadrículas de 50 por 50 metros de forma aleatoria en la isla de Tenerife. Este hueco que ven aquí en el medio representa la cota de los 1000 metros. Es decir, nosotros trabajamos nada más que de 0 a 1000 metros, porque sabíamos que la distribución de las parcelas cenicientas está principalmente ligada a esos primeros 1000 metros de altitud, ¿vale? Por encima de ahí puede haber algún nido, pero las densidades son ya muchísimo más bajas. Tras el trabajo de campo, conocemos actualmente más de 1900 nidos y los tenemos ploteados, que son estos que se muestran aquí en el gráfico de la derecha. Con esta información hemos hecho mapas, modelos de distribución de especies, como máximas, que pueden ver en este mapa. En este mapa pueden ver los lugares, las cuadrículas donde hay parcelas cenicientas brillando, y en la gama de colores pueden ver la mayor probabilidad de presencia de la especie. El azul es que es 0 y en rojo las zonas con mayor probabilidad de reproducción. Aquí en la parte derecha es justamente lo mismo, pero cambiado el color a una escala de 4 colores, donde el rosa indica de 0 a 25% de probabilidad y el rojo más intenso indica aquellas cuadrículas que tienen más del 75% de probabilidad de ocupación. Bueno, pues con esto podremos hacer un mapa de distribución de la especie y además podremos intentar cuantificar el tamaño de la población. Pero hay otra forma de cuantificar el impacto de la contaminación lumínica en la población. Y podría ser utilizando la información que ya sabemos de la literatura. Perdón, para conocer el número de parejas reproductoras. Podemos utilizar la información que ya tenemos de la literatura. Aplicando esta fórmula, estos valores, los valores de que ya sabemos, Rodríguez y colaboradores estimaron que el 14% de los pollos eran afectados por la contaminación lumínica. El Cabildo de Tenerife, a través del Centro de Recuperación de la Taonilla, dice que rescata en el año 2017 2.580 pollos, y además, gracias al proyecto Luminado, sabemos que el éxito reproductor de la especie en la isla es alrededor del 53%. Con esta información, podemos decir que el tamaño de la población en la isla de Tenerife es alrededor de 30.000 parejas. Y ojo, esto es un valor muy alto comparado a lo que se había dado previamente, que lo he representado aquí abajo. En el año 2007, Lorenzo, en el libro del Atlas de las aves reproductoras de Canarias, daba un dato de entre 2.000 y 3.000 parejas reproductoras. Años más tarde, nosotros mismos dimos un rango de entre 8.000 y 16.000 parejas reproductoras. Y ahora parece que el proyecto Luminase arroja un número mucho mayor. Esto hay que tomarlo con cautela, pero lo juntaremos con la información que tenemos de los modelos de distribución y veremos si esas cifras se aproximan o no a los valores que nos salen siguiendo los modelos de distribución de especies. El punto número 2 de aquella lista de 5 puntos que habíamos compilado son los límites de contaminación lumínica y las distancias a las fuentes de las luces. El proyecto Luminaves ha intentado arrojar cierta información acerca de este aspecto, y para ello lo que hemos hecho es marcar aves con GPS, con unos dispositivos especiales, que lo que hacían era enviar la información de forma remota, con lo cual no hace falta recuperar el ave para descargar la información. Y esto es muy importante, puesto que nos permite obtener información de todas las aves que lideramos, ¿vale? Lo que encontramos fue lo siguiente. Estas aves que habían sido afectadas ya por la contaminación lumínica las subimos a un lugar de liberación experimental en la zona del sur de Tenerife y seguimos su vuelo hasta el mar. Podemos ver como otra vez las zonas turísticas de la Oficina de las Américas y de las galletas en Montaña Amarilla son 2 zonas calientes donde las aves se ven atrapadas. Y aunque algunas de ellas lleguen al mar, pasan por ahí y por tanto son susceptibles de esas aves. Y por último, el último punto fue conocer o investigar más acerca de las características de las luces que afectan a las aves. Para intentar responder a esta pregunta, hemos tomado los datos de una asociación que se llama Marinas, Asociación Svenja por la Naturaleza, que trabaja en el Valle de Weimar rescatando aves desde el año 2010 al 2019, es decir, 10 años de datos. Lo que hemos hecho con esos 10 años de datos es mapearlos, como pueden ver en esta figura de aquí, a la izquierda, mapearlos en la zona y calcular un kernel de distribución. Después hemos generado puntos aleatorios siguiendo ese kernel de distribución, aquí los puntos azules, y los puntos verdes hemos mostrado puntos aleatorios en el área de estudio. Además, hemos utilizado una imagen de la Estación Internacional Espacial y siguiendo la metodología de Sánchez de Miguel, hemos intentado determinar cuáles son los puntos más difíciles, los puntos más calientes, o sea, cuáles son las fuentes de iluminación y caracterizarlas, es decir, si son amarillas, si son blancas o qué tipo de luces son. Lo que hemos encontrado en estos programas, en estos box plots que se muestran debajo, es justamente los box plots rojos son las pardelas. Podemos ver como los lugares de hallazgo de pardelas son todos más iluminados para los 3 colores, para el rojo, para el verde y para el azul. Pero es que además podemos ver que hay una diferencia importante con las zonas aleatorias y los puntos aleatorios siguiendo el kernel. Incluso siguiendo la distribución del kernel, las pardelas parecen estar en lugares más iluminados. O sea, haciendo una comparación entre los azules y los rojos, que son los lugares de la pardela. Esto es muy importante porque hasta este momento había una cierta baja información acerca del tipo de luces que afectaban. Se cree, se estima y se ha visto que las luces blancas son más dañinas, pero bueno, nosotros queríamos testarlo con datos ya que se habían hecho con un dataset más largo, ¿vale? Y bueno, después de toda esta charla, ya para finalizar, yo estaría muy agradecido si después de toda esta charla ustedes llevaran 3 ideas clave a casa. La primera de ellas es que la contaminación lumínica es una fuente de mortalidad para las aves marinas que crían bajo tierra, y es muy importante para ellas. La segunda es que es un problema bastante viejo, pero que está lejos de ser completamente entendido. Y la tercera es que necesitamos más investigación para poder diseñar y mitigar los impactos de la contaminación lumínica y para diseñar acciones que promuevan una mejor iluminación. Y por último, y para finalizar, me gustaría dar las gracias a toda la gente que ha colaborado conmigo, que son muchas personas. Primero me gustaría dar las gracias a los centros de investigación a los que pertenecidos, al GOHNIC, al Grupo de Ornitología e Historia Natural de las Islas Canarias, a la Estación Biológica de Doñana, del CSIC, y a Phillip Island Nature Park de Australia. También me gustaría dar las gracias a la gente con la que colaboramos activamente en la colecta de información y de datos, principalmente el Cabildo de Tenerife a través del Centro de Recuperación de la Tabanilla, también a TINA, también a DIRRI de Baleares y a la Red Española de Estudios para la Contaminación Lumínica de España. También me gustaría agradecer a las organizaciones que han financiado parte de estos estudios y por supuestísimo al proyecto Luminaves y también a SeaBirds por liderar este proyecto. Aquí debajo les dejo mi contacto y también más información acerca del proyecto Luminaves y de mi página personal acerca de Ecolife for Seabirds. Muchas gracias a todos.
Resultados muy interesantes y espero que aunque el proyecto Luminaves termine ahora, que podamos continuar trabajando en esta temática y en caso de Madeira saber un poco más sobre la parte ecológica y más relacionada con las aves, los impactos, que es algo que aquí aún no hemos empezado. Os acuerdo a todos que pueden después poner vuestras cuestiones en el chat de esta reunión y Airam a aclarar algunas dudas y compartir más información con nosotros. Muchas gracias.
Gracias. Muchas gracias a todos. No se corten, hagan preguntas. Gracias.